testing, then ethical and regulatory considerations, future prospects, then advanced exo. I will talk about a short overview of advanced exo, then Koshika's contribution in advanced cells products, and then conclusion and question answer round. So the cosmetic market. So in 2023, the global cosmetic market was valued about 422.15 billion USD, which is expected to increase by 635.68 billion USD by 2030, with the CAGR of 5.25%, which is the forecast for 24 to 2030. Despite the global recession, through which the global recession is going on in global recession, despite this, beauty and personal care market is continuously increasing, and people are more focused to use organic and natural products. So as you can see, people are more now people don't want to use chemical based skincare products or something like that. They want to use organic natural products, and the product natural product should be backed by scientific reasons, that why this skin product is working like this, and all the skin cosmetics should be scientifically backed. And you can see in the graph that global anti-aging market is also one of the share in the cosmetic industry. And not even women but men are also focused to use these anti-aging cosmetics. You can see 19% of women is using anti-aging cosmetics, but men are also 81% of the share. And the main market for now is Asia Pacific, and North America market is expected to grow in this year, in all these years, which is now behind Asia Pacific market. So then I want to discuss about structure of human skin. So human skin is made up of two layers. First is epidermis and second is dermis. So epidermis is very thin layer, and it acts like a barrier. So it resists any bacteria or any fungus to get inside the skin. It function as a barrier to the skin. And the lower layer is dermis. So dermis is much thicker and a lower part of the skin. And as I said, epidermis is barrier, so it contains keratin. Keratin is a protein which resists the bacterial entry. Then it prevents loss of fluid and electrolytes from the skin. And dermis is well, and epidermis is not vascularized. It's a keratinized epidermal layer. And dermis is well vascularized. It contains receptors for touch, temperature, and pain. And as I said, epidermis contains different types of cells like keratinocyte, which contains keratin. Keratin is a skin protein. Then melanocytes. Melanocytes contain melanin. Melanin is a skin pigment. So the skin color across the globe depends on this melanin. If some person is in the skin contains more melanin, then he will be or she will be more darker as compared to the person containing less melanin. Then other cell type in epidermis is Langerhans cells. It's a macrophage of the skin, like immune cells of the skin. It protects the skin from bacterial and other cells. So, what is skin tissue engineering? So, skin tissue engineering is made up of three components. One is cells, other is scaffolds, and the third part is growth factors, signaling factors. So cells are, for skin tissue engineering, you will require keratinocytes, melanocytes, mesenchymal stem cells, then dermal fibroblast. Scaffolds is nothing but the ECM, extracellular membrane. So in vivo, skin contains extracellular membranes, like it work as a cushion to the skin cells. So it can be hydrogels, it can be porous scaffolds. Then you can electrospun these fibers like collagen or gelatin, all these ECM materials. Then to culture these cells in vitro, you need some signaling molecules like some chemicals, growth factors, which are required to grow these skin and maintain these skin cells in vitro. So this is the basics of skin tissue engineering. And why we are doing this? So we are mimicking the in vivo condition, that is the human body condition in the plate. So we are in vivo culturing the skin in the plate. What are the applications of in vitro skin models? So we can make three types of skin models. First is engineered skin tissue, which I will discuss in my later slide in details. Then skin organoid, which is made up of induced pluripotent stem cells, and skin on a chip. It's like a microfluidic chip where you culture all the types of skin cells, and the skin chip will be ready for your testing. So, and all these in vitro skin models are used for whether for drug screening for skin diseases like psoriasis, SLE, and other inflammation in the skin. Then cosmetic testings like sunscreen, skin whitening, anti-aging cosmetics, all these things. And in 2021, the FDA Modernization Act has compulsate the use of these skin models, and they want animal models to be reduced for toxicology or any safety studies. So now all these model skin models are in the market for cosmetic industry, and for cosmetic testing. The in vitro skin models are, the demands are increasing for cosmetic testing. So how you will make the engineered tissue? So for autologous, if what is the advantage of autologous human? If you want to test some cosmetics which you want to test for aging purpose, so you can take the skin of aged person. The skin will be aged, so it will mimic the aging tissue. If you want to mimic the young person's skin, so you can take 16 years old or 20 years old person's skin, it will be young skin. So you can take the skin biopsy, and you can isolate easily from their keratinocytes, melanocytes, fibroblasts, Langer cells, endothelial cells, all these types of cells. And then you have to just mix these cells and culture it in the scaffold. Or if you don't want to use a scaffold, then the fibroblast has this capacity to self assemble, and when the fibroblast make the sheet of the cells, so it forms a ECM also. And in that ECM, other cells like keratinocyte, dermal fibroblast, macrophages, all the cells will get attached, and it will form a engineered tissue. And if the skin is vascularized and it contains immune cells also, then it is the best model to mimic the any skin disease or any engineered tissue for in vitro drug screening. So this is the evolution of in vitro skin models. So first, people were using inert membrane, like just on the plastic surface. They used to culture this keratinocytes and other cells. Then they started using synthetic matrix like vitronectin, any other synthetic materials like polycaprolactone and all. And then they started seeding on them the basal and differentiated keratinocytes, and then they started mixing co-culture of fibroblast, basal keratinocytes, differentiated keratinocytes. Then they started in the C figure you can see, and then they started self-assembled matrix. As I said, the fibroblast forms a layer, and it secretes natural ECM. So that is the most evaluated matrix for skin tissue engineering. And then they have added this basal keratinocytes and differentiated keratinocytes. Now the most advanced model is where they have added all these immune cells also, like dendritic cells, like lymphocytes, macrophages, granulocytes, and vascularization also they have introduced in the latest model. And other sheet, adipose tissue sheet also they have added in this advanced in vitro skin model. So, how you can characterize these in vitro skin models? So, there are some protein markers like filaggrin, involucrin, loricrin, aquasporin, transglutaminase, keratin 10, keratin 5. Then there are some dermal, epidermal junction markers like collagen 4, laminin, BP antigen, and proliferation marker like Ki67. This all will say that how functional your skin model is, and if it's expressing all these markers, then your epidermis and dermal layer is well intact, and it's expressing all the protein markers, and your skin tissue engineered sheet is functional, and the cells are proliferating or not. So Ki67 marker will tell you the cells proliferation status. Second, other thing is skin organoid. So this is the latest model where the induced pluripotent stem cells, iPSCs, you can differentiate it towards skin cells by introducing different growth factors and signaling molecules. Then in the plate after 28 or 30 days of differentiation, you will see the formation of skin organoids. And when you will characterize these skin organoids, then on the right side you can see how it's mimicking the adult skin and its structures. So you can see it's expressing keratinocytes, keratin 5, collagen 3, then melanocytes marker also, melan A. All these markers were get expressed in skin organoid. So this model is also a good model to test cosmetics. Then other model is like 3D bioprinting. So this can be used for two applications. First will be for drug testing and disease modeling, and other for treatment of patient. So in this, what you have to do, either from the patient or from the healthy volunteer, you have to take skin biopsy, and from there you have to isolate keratinocyte, melanocytes, and dermal fibroblasts. Then you have to mix all these three types of cells with a suitable bioink. Bioink is nothing but extracellular matrix which is required for skin cells to grow in vitro. Then you have to mix your suitable bioink and you have to print. So when you will print these 3D skin cells, then skin construct will be formed, and it can be grafted in the patient, or it can the skin construct can be used for disease modeling or drug testing. So these are some of the bioink which are used for skin bioprinting widely. First is collagen. As we know, collagen is one of the most prominent extracellular matrix in the body. Then second is gelatin. Then third is fibrin, alginate. Alginate, chitosan. Chitosan is a found in fungus. So this is not the natural body polysaccharide, but it's widely used for skin tissue engineering. And then there is hyaluronic acid. So all these bioink has their own advantages and disadvantages. So, what are the advantages of 3D bioprinting skin models over animal models? As I already mentioned, that in 2021, FDA has made this rule of FDA Modernization Act. So now they want for all the drug screening and other purposes, animal number should be reduced. If you are using any animal, then it should be stress free for the animal. And third, they want to replace it completely. They want to replace animal models with 3D models like organoid, 3D microfluidic chips, or any engineered tissue. So, what are the disadvantages of a skin? That you don't have options of aged skin or young skin. whatever skin you will get in the animal, you have to use that. And it's not mimicking the human physiology, it will mimic the animal physiology. You will not get any young skin or aged skin. Another limitation will be the skin disease condition. If you want to study psoriasis, which is a inflammatory skin disease, so in mouse you will not able to mimic the disease human diseases, and then there is a storage problem. And in other case, animal facility maintenance is also costly. So in the other hand, if you will use 3D printed models, so you can get patient cells, you can mimic human physiology. The skin aging also you can mimic by taking the skin from the aged patient or aged healthy volunteers. Or if you want to study young skin, then you can take a skin biopsy from young healthy volunteer. And these skin models are easy to store. You can store it at -80 or -20 temperatures, and it's ideal to study any rare disease also. Then these are some commercially available human skin models, like reconstructed human epidermis, which is available from L'Oreal, Mactec Corporation, Epiderm, Kera Skin, all these, and they are validated for different skin testing, cosmetic testings. And you can see different scaffolds have been used for all these commercially available human skin models, like collagen, polycarbonate. Then these are some pigmented RH reconstructed human epithelium, in which they have added melanocytes also to study skin whitening reagents. So melanoderm, then human skin equivalents are there, which is also used for multiple applications. So these are the characterization of in vitro skin models, where they have studies different markers like trans-agglutinase, keratin 10, laminin 5, Ki67, all these. So this is the paradigm of cosmetic testing methods. So any cosmetic ingredients, first you have to check whether it's carcinogenic or not. So for this, you have to perform genotoxicity study. Any cosmetic product should not cause cancer. First criteria is to test that. Then it should not cause any skin sensitization or skin irritation. Then you have to check eye irritation study, then photo irritation. If you have applied any cream or any ointment, then if you are going in sunlight, then it should not be irritating. Then you have to check its absorption study you have to perform. Then you have to fix a margin of safety for using all these skincare. So this is one of the skin ethic, which skin reconstructed epidermis, which is available in L'Oreal cosmetics. So skin irritation study is performed on these small strips or on the skin discs also. So here they test whether any cosmetic ingredients is causing any irritation or not. In downside figure, you can see in vivo DAS skin test, where the skin irritation test was performed on rabbit also. So here you can see the redness. So whatever redness is formed, it will be calculated uh, by the inflammation marker and MTT, which is a cytotoxicity assay. So the irritant percentage will be calculated on the basis of that. Then second is in vitro permeation test. So any cosmetic ingredient which are used should permeate inside the skin. As you see, resorcinol is chemical which is generally used for acne, dermatitis, eczema, psoriasis, and other skin disorders. So you can see how it's crossing the skin disk, and when the resinol will cross this dermis, so this skin disk is cultured on the trans well. So you can see this yellow small molecules are going down. So this will be calculated by Raman spectroscopy and other permeation study. and it will calculate the time, how much time the drug is taking to going inside the permeabilizing inside the skin and all. Then there is apicular eye irritation test. So apicular tissue is keratinized ocular tissue. So in this, the tissue, skin tissue, will be cultured on the transwell, and the cosmetic will be introduced on that disc. So if the cosmetic is causing any cell toxicity, and it's more than 60% of cytotoxicity, then the chemical is non-irritant. And if it's more than 60%, then it's irritant. Here in right box, you can see how the cosmetic is causing dead tissue and viable tissue. So this can be performed by MTT assay. There is in vitro phototoxicity test, so where the skin tissue was first treated with cosmetic ingredient, and then UVA light and UVB light was exposed on these skin disc, and then phototoxicity or photo irritation study was assessed by MTT assay or LDH. LDH is also a very good cytotoxicity study assay. Then, as I said, any cosmetic ingredient first should be added for genotoxicity test. Genotoxicity test means the cosmetic should not form any cancer. So in this also, we people are generally using a comet assay or micronuclei assay. And if for genotoxicity assay you are doing cytotoxicity study, so if more than 50% cells are dying, means the ingredient is highly cytotoxic. If 30% cells are dying, then it's intermediate cytotoxic, and 10% cytotoxicity is okay. So this is low toxicity. And your micronuclei, if it's more than 40% nucleus has become binucleated, then the your ingredient is cytotoxic. These are some registered artificial skin products like AP Skin from L'Oreal, Mediderm from Mattec, Skin Ethics from Skin Ethics France, APCS from Henken Germany, AP Ocular again from Mattec USA, and all these skin products has different applications, like some can be used for phototoxicity, some can be skin irritation, some skin corrosion, and like skin whitening. If you will see, skin whitening, skin corneal permeabilization, all these are different types of skin models available for cosmetic testing. These are some more. So melanodome is one of the advanced 3D in vitro co-culture skin model which contains keratinocytes and melanocytes. It's available in MagTech USA. So it's what happened in these cells, that NHM is normal human melanocytes. So in vitro, in co-culture conditions, they undergo melanogenesis. That it means that they form melanin inside the cells, and melanoderm is produced with it's a serum free media and without any artificial stimulation, which it does not contain any artificial ingredients. And it's cultured, the skin insert is cultured in air liquid interface, and generally it is used for skin lightening and self tanning agent testing. So this is how how cosmetic is testing for skin whitening. So if the skin is forming more tyrosine and melanin, it means dark skin. And if the skin is producing less tyrosine and melanin, then the skin will be lighter. So this is how melanoderm works. So in this testing, 2% kojic acid, which is a positive control. Kojic acid is generally used in ointment for skin lightening, and magnesium ascorbate Phosphate is also one of the skin lightening chemical. So these two are positive controls, and negative control is water. So then three tests have been performed for dark spot corrector 1, 2, 3. So you can see at day 14 how all these three correctors, spot correctors, are working. According to me, corrector 2 is working more efficiently. And how you will analyze these tests? So for this, you have to perform tyrosine production assay, like melanin assay, macroscopic photos where you will see the decrease of melanin synthesis, and other histological imaging you can do. This is molecular mechanism of skin aging. As we age, our skin, even the whole body get age, but skin is more visible. So we can see the skin aging more prominently in the body. So what happened during aging? So during aging, your epidermal thickness is decreasing, your number of melanocytes are decreasing, then melanocytes are more melanocytes are getting died, number of Langerhans cells, that is the macrophage of the skin, is getting decreased, moisturizing factors will decrease, skin hydration will decrease, then there will be more MMP formation, TIMP formation, extracellular matrix like hyaluronic acid and all these will decrease in the skin. And if you are getting more exposed to light, then the skin aging will be more prominent, and early it will cause more early skin aging. So how you will test the anti-aging efficacy of the ointments? So you can perform multiple tests like antioxidant activity, anti-inflammatory, anti-glycation, anti-tyrosine, collagenase, anti-hyaluronase studies. So this is one of the case study of fenugreek seed. So fenugreek seed is anti-collagenase. So applying fenugreek on your skin will make you look more younger. There is gala green. Gala green is a has anti-inflammatory and antioxidant properties. So if you will apply this plant extract of gala green, you will look more younger. Then exosome. Exosome is one of the most advanced therapy for skin regeneration, and it's a regenerative. As I said, the people are now more focused on chemical-free and scientifically driven cosmetic products. So people are now more demanding for this exosome therapy, where the exosome, how these exosomes act for skin regeneration. So it's causes an angiogenic ability, it increases, then collagen synthesis it increases, then it regulate any inflammation in the skin. What is exosomes? So exosomes are the nano bodies which are secreted by the cells. So some of the sources for exosomes are like menstrual blood, plasma or blood. Then some MSCs, exosomes can be secreted from umbilical cord, mesenchymal stem cells, bone marrow, adipose tissue, amniotic MSCs and all. So it affects mm, it regulates endothelial cells, macrophages, all these keratinocytes. It increases proliferation and migration and reepithelialization of keratinocytes. Then in fibroblast also it causes increase in the proliferation, migration and collagen deposition. So this is how exosomes works. Then exosomes is also effective in photoaging. As you can see how this photoaged skin has been rescued by exosomes. Then this is how exosomes are increasing elasticity of the skin and decreasing the wrinkles on the skin by increasing collagen, elastin, fibroblast, and keratin in the skin. Then exosomes are also helpful in wound healing. Like if you have some chronic wounds or burns, very severe burns, then these exosomes will go and act, and it will impair the skin. Then a skin flap, if some skin flap is tagged on your skin, then the exosomes will act beautifully there. It will remove the scar, it will regenerate your skin, it will remove the pigments, and it will also help to grow skin on hair on the skin flaps. Then SLE is a autoimmune disease, and it causes lacy pattern under the skin. So exosomes are also effective on SLE. Then other example is psoriasis. Psoriasis is also an inflammatory disease. It's a skin inflammation disease where your skin becomes very irritating, and inflammation and dryness of the skin. Your skin will become dry. So exosomes has been shown effective on psoriasis also. Then other disease is atopic dermatitis. It's a skin inflammatory disease. So exosomes also works beautifully on atopic dermatitis. Then scar removal, exosomes act beautifully on scars. Then vitiligo is skin. Your skin will lose melanocytes or melanin. So the regular application of exosomes can treat vitiligo also by decreasing some inflammatory markers. And exosomes is also used for systematic sclerosis. Systematic sclerosis is also one autoimmune disease. So it has shown, exosomes have shown a beautiful results in systematic sclerosis patients. Then ethical and regulatory compliances. So I will discuss about how these engineered tissue can be, how we can make engineered tissue in the lab. So first we have to take ethical committee approval for the project. uh, and then we have to, from the donor, we have to take signed consent. We have to protect the donor's privacy and confidentiality, and we can even anonymize donor's name also. And in the lab we have to follow GLP, good laboratory practice. These are some of the national regulations. In USA it's FDA, in Europe, it's a European Medicines Agency, and in India, it's a Central Drugs Standard Control Organization (CDSCO). So these are the some regulatory bodies which take care of the ethical and regulatory compliances all over the world. And in India, it's a CDSCO. So again, the advanced cells group is offering this advanced exo, which is for skin regeneration and hair regeneration. So a skin regeneration kit is making the skin brighter, and even it's control dark spots and pigmentation. It's helpful to manage the skin. And hair regeneration kit is helpful to boost your hair follicle, and it helps to reduce hair fall also. Yeah, thank you. Any questions? So this session is open for questions now.